G'day and welcome guys to the fifth part of this presentation. My name is Dr. Buddhamana Niyakara, respiratory and sleep physician at Orange Health Service, a clinical lecturer at Charles Sturt University, clinical lecturer at University of Sydney. So, we've discussed that oxygen is vital in aerobic respiration. We've talked about how oxygen gets to the tissues by virtue of coming from the environment through the nose, humidified air, into the alveoli, diffusing down its partial pressure gradient into the pulmonary circulation, carried by bulk flow, pumped through the heart into the tissues where oxygen diffuses into the mitochondria to produce ATP. We realized that there was a problem in that oxygen in blood is poorly soluble by itself and it must be carried by a carrier protein called haemoglobin. We then spent quite a bit of time discussing how haemoglobin carries oxygen. We talked about deoxyhemoglobin and oxyhemoglobin and how hemoglobin displays a property known as cooperativity for oxygen. We discussed some of the modulators of hemoglobin's behavior, such as pH, carbon dioxide levels, temperature, and 2,3-DPG, and how a hemoglobin has physiologically adapted to its needs. What we're going to talk about now is we're going to introduce the red blood cell, and we're going to talk about how the red blood cells carry hemoglobin and some of the structural modifications that allow it to carry hemoglobin and in fact deliver the oxygen. So let's move on. So this is an idealized picture of the red cell, and what we can see with the red cell is that it's got a really interesting shape. It's what we call biconcave. I'll just draw that here, but I might just need to change the pen color to blue to make it a bit clearer. You can see that's biconcave. It's fatter at the sides, skinnier in the middle, and it's got a bit of a donut shape. Now, if you can imagine, for hemoglobin, so for red blood cells to carry a bucket load of hemoglobin, we probably need to give some of the contents of the cell away. So therefore, in order to carry a truckload of hemoglobin within the red cell, red blood cells have given away their nucleus and all their organelles. And that's important because remember, it's the nucleus that synthesizes the mRNA that gets translated into proteins. Without the nucleus, we really cannot synthesize proteins. Without endoplasmic reticulum, without the ribosomes, we cannot translate proteins. So red cell has to cope without all of these things. It can't make new enzymes, can't make new functional proteins. The other thing to note is that because red cells need to absorb the oxygen that dissolves into the blood, for it to be loaded onto hemoglobin, it should have a high surface area to volume ratio. It should be exposed to as much blood as possible through its membrane. And this is one of the reasons why it adopts this characteristic biconcave donut-shaped appearance. It greatly increases the surface area to volume of the red cell to obtain oxygen from the environment and load it into hemoglobin to cause oxyhemoglobin. Furthermore, the biconcave structure also helps the red cell to deform easily, and that's important because if you think about it, in the tissues, the capillaries that lead to these tissues are very, very small. So the red cell has to twist, has to be deformed. Remember that electron micrograph of the pulmonary circulation? We saw that red cell kind of bending over itself. It can't do it in other configurations. It really is this biconcave configuration that enables a red cell to do that. And one of the ways that the red cell is able to do that is because it's got this complex structure of vertical and horizontal cytoskeleton elements, mainly made up of a protein called spectrin. And one of the physiological importance of spectrin can be seen in a red cell inherited deficit called hereditary spherocytosis. Now, you don't need to know really anything in depth about this. I'm sure that we will cover it in our hematology block. But essentially, to illustrate the point, one of the defects, there are many, but one of the defects in hereditary spherocytosis is that it lacks some of the vertical cytoskeleton spectrin elements. And because it lacks that vertical stability, what happens is that the red cells start to shed membrane, and when they shed membrane, they shed surface area. And when they shed surface area, the red cells go from being biconcave like this into spherical structures. And when they become spherical structures, they reduce their surface area to volume, so they're inefficient at obtaining oxygen into the red cell to be given to hemoglobin. And also, they're not as deformable. And one of the things that happen is that when it passes through the spleen, which is a blood's filtration system, these spherical red cells, hence the name hereditary genetic spherocytosis, they get stuck in the splenic microcirculation, they get taken up by the spleen, and they get destroyed. One of the ways of treating this is to take the spleen out. If you take the spleen out, there's nowhere for these cells to get destroyed. Now, let's talk a little bit about the enzymes involved in red cell metabolism, because I think, although I wouldn't say this is examinable, not certainly for this lecture, but it does illustrate a point. So remember, red cells lack nucleus, they lack organelle, so they don't have mitochondria. If you don't have mitochondria, remember, there is no aerobic respiration. Doesn't exist. So we can only do it through anaerobic respiration, which is essentially glycolysis. Now we'll see what happens to glucose. So glucose can be metabolized through glycolysis, and it produces 2 molecules of ATP. We learned a bit about that in one of the previous parts. Also, one of the fundamental things that happens in red cells is that at this level, at the level where the glucose has been phosphorylated using something called a hexokinase, we create glucose 6-phosphate, and that can be shunted through what we call the pentose phosphate shunt to produce different other sugars. Now, the fundamental importance of the pentose phosphate shunt is that it is the only way for the red cell to produce NADPH. And the reason why NADPH is so important is that this is, again, an electron-rich molecule, much like NADH, much like FADH2, which we learned about very briefly in a talk on aerobic respiration. But NADPH is fundamental in that by becoming oxidized to NADP, it's able to reduce glutathione from GSSG through to GSH. And reduced glutathione, or GSH, is fundamental antioxidant. So it's able to donate electrons and get rid of potent oxidants. Now, one of the things that can happen to red blood cells in patients suffering from G6PD is that they have a deficiency in the pentose phosphate shunt, and so they cannot form NADPH. Let's just say the red cell has an oxidant hit from some kind, some drug, fava beans can do it, or an infection, some kind of oxidative stress. Now these oxidants, they damage hemoglobin, they cause hemoglobin to associate with each other because the tetramers form what we call disulfide bridges, which occurs due to oxidation toxicity. Normally what would happen is that the reduced glutathione in the form of GSH smashes these bridges apart and the hemoglobin molecules deassociate. But if we don't have this, because we can't create GSH, because we don't have NADPH, because we lack the enzyme glucose 6 phosphate dehydrogenase due to G6PD deficiency, what happens is that oxidative damage goes unhindered, causing disulfide bridges to form on the hemoglobin tetramers, causing hemoglobin molecules to precipitate. Hemoglobin molecules, to when they precipitate, actually destroy the red cell membrane, because they destroy the cytoskeleton, and that causes vulnerability and basically hemolysis, breakage of the red cells. And what you can see here is a blood film taken from a patient that has G6PD deficiency, and what you can see here, all these kind of nuclear bodies, they're called Heinz bodies. And essentially what's happened in this patient is that through oxidant damage, it's caused this disulfide bridges to occur between hemoglobin molecules, causing precipitation, and this is what you can see through microscopy, precipitated hemoglobin. And remember, precipitated hemoglobin destroys the internal structure of the red cell, damaging it, and that makes it susceptible to hemolysis. Now I hope that all makes sense.